Hello friends, welcome back to my channel Xgene and proteinX. In this tutorial video, I'll discuss about the overview of the spike protein mutation and the immune escape that induced by the spike protein. So as you see in this figure, in the left side figure, it's the schematic diagram. It's representing the infection by the SARS-CoV-2 through the S2 receptor. So when the SARS-CoV-2 it infect any host cells through the S2 receptor, then it get entry this SARS-CoV-2 virus inside the host cell. So after that, the immune response that arised inside the host cells. So what happened there? There are a lot of immune cells like CD8 positive T cells, CD4 positive T cells, memory T cells, and anti-SARS-CoV-2 antibody. These are all generated inside the human host cells. So as you see here, there are a lot of immune cells as well as the anti-SARS-CoV-2 antibody. So the memory T cells, it protects against this kind of infection when this kind of SARS-CoV-2 it infect in future. And also the CD8 positive T cells, it are also known as the cytotoxic T cells. So when any infected cells is recognized by the CD8 positive T cells by some crucial mechanism, then these cytotoxic T cells it destroy this kind of infected cells. And also the CD4 positive T cells, it also known as the helper T cells. So it helps to the CD8 positive T cells and also other immune cells to accumulate near the infected cells. And also the anti-SARS-CoV-2 antibody, it reduce the infection, right? So this is the natural immunity and natural immune response that induced by the SARS-CoV-2. So I would like to mention one more thing, that over time the number of these immune cells and the antibody response it declined. So as you see here in this figure, that over time the number of this kind of immune cells it reduced. So whereas this kind of immune response it reduced as well. So in this case, this kind of repeat infection can be caused, and also some other infections may arise. So in general, like this kind of natural immune response are largely protective for at least 6 months post infection. All right. So now I wanna show the mutation part. Then how this kind of mutation it impacts. So here as you see in this figure, because in the earlier slide, as I mentioned, that the antibody that generated against this kind of the RBD and also NTD, so this kind of antibody are very specific. They can recognize this kind of specific kind of antigen or protein, and then they bind with this antigen and then destroy their efficacy. All right. So here you see, as you see, this is the spike protein, and the different part of the spike protein. These are the S1 part, S1 subunit, and this is the S2 subunit. So in the S1 subunit, it contains the RBD and NTD. So this kind of antibody are generated against this RBD and NTD. So as you know, the RBD is the receptor binding domain and NTD is the N-terminal domain, right? So what if this mutation occurs in this site? So if such kinds of mutations it occurs in this kind of sites, then this kind of antibody they cannot recognize very properly this kind of antigen. Thereby the immune response or the activity of this antibody it reduced. All right. So here you see the different kind of mutations I mentioned here. These are very much crucial mutation but as of now there are a lot of mutations is already observed, but out of which there are some mutations are very much crucial and very much important for this kind of SARS-CoV-2 virus, for their more infection and more transmissibility and also other actions. So here I would like to mention that each of these mutations and what the impact of this kind of mutations. So here as you see this L452R, these mutations are normally it found in the delta, epsilon and kappa variants, and it resists the neutralization by monoclonal antibody and vaccines. And also it has this kind of mutation, it also has the greater affinity binding to the S2 receptor and promote viral replication and infectivity. So these are the very much common mutations. So also the N501Y, this kind of mutation also it observed in alpha and gamma variant, and this kind of mutation it reduces the virus sensitivity to the monoclonal antibody and vaccine. And also it was found that in the in vitro study, so they did some the study on pseudovirus, and it has been observed that this kind of mutation it increase the viral infection rate 5-fold compared to the normal one. All right, so this is also very much crucial mutation that that commonly occurs, and also the infection rate also is higher in this kind of mutations. So next move on to the another mutation. This is the E484K, these mutations, and this is also found in the alpha and gamma variants. And this kind of variants, it mildly increase the affinity of the RBD to the S2 receptor. But here also another one important thing that this kind of mutation, it helps these mutations for the engagement of the receptor to the S2 receptor. All right, so this kind of mutation it enhance this mutation for engagement to the receptor, S2 receptor, and this is the longer time they can engage to the S2 receptor. And another important feature it found in the E484K that is very much different than compared to the other variants. Like this kind of infection it found that in the vaccinated individual and with this kind of carrying variant. All right, so this is very much important features of this mutation. And another common mutation, that is a D614G, and this variant it was very common, and it was the earlier variant. So it was found just after the outbreak, within 6 months. This kind of common mutation it was found, and it was very much common at that time. And the Omicron variant, this is the latest variant, and this variant it has around a lot of mutations, like 30 amino acid changes it found, and 3 small deletions, 1 small insertions, and in the spike protein. And so because of this kind of lot of mutations, it leads to this kind of virus, it is higher transmissibility rate and lower the deadliest disease. So this kind of virus is not that much fatal, not that much deadly compared to the other variant, but in this case the transmissibility is higher in this Omicron variant. So this is the all of the important and crucial mutations that I already talked. So let's move on to the next slide. This is a lot of mutations that I summarized here. There are a lot of mutations. So as you see in this figure, that the NTD, N-terminal domain, this is the RBD domain, and this is the subunit 2. So this is the subunit 1 and subunit 2. So this is the receptor binding domain and the N-terminal domain, right? So here as you see, this is this kind of mutations it observed in this NTD part, and this kind of lot of mutations is also observed in the RBD part. So in the RBD part, we know that RBD is mostly important for the binding with the H2 receptor. So NTD, it has the main function of the NTD is fuse in the cell membrane, and the RBD, it bind with the H2 receptor. So this is for the RBD is for the higher infection. If the mutation it happens in the RBD, so it increase the infection rate and the transmissibility. So as you see in this figure, there that this kind of monoclonal antibody, this kind of so here you see this all these mutations. This kind of mutation shows that it promotes resistance to the neutralizing monoclonal antibody. So here there are lot of this kind of mutations is found, and these all kind of mutations, these are it can promote the resistance to the neutralizing antibody against the RBD. All right, and also this black spot, these kind of things, these are for the vaccinated individuals. So this kind of mutation, it promotes the resistance against the vaccinated individuals. So this all kind of mutations are very crucial, and for mostly for their higher the infection rate and transmissibility, and reduce the neutralizing the monoclonal antibodies and the vaccines. So this is all about this the spike protein mutations. So I hope this video will be helpful, and if you like this video, kindly hit the like button, share it, subscribe my channel, and if you have any queries, kindly write in the comment section. Thanks.